Hey guys, it's Professor Tiffany Martin with Medical Microbiology, and we are in Chapter 16, Adaptive Immunity. And let's go back and talk about some T cells again. You guys told me that T cells were produced in the bone marrow and they matured in the thymus, which is where they got their T part of their name from the thymus. So what happens in the thymus? What happens to make them mature in the thymus? So this process is called clonal deletion, and that's what we're going to talk about next. Okay, so T cells mature in the thymus and they undergo clonal deletion. Just for your reference, B cells also undergo clonal deletion, but they do it in the bone marrow, right? B cells mature in the bone marrow, B for bone marrow. So let's talk about the T cells first and the process that they do in the thymus, but know that the B cells do a similar process in the bone marrow. All right, the purpose of clonal deletion is to make sure that all of these T cells are actually able to do their job. So we want T cells to be able to do their job. We also want to make sure that they don't attack our own body cells. Again, these are the big guns, right? The adaptive immunity, the T cells, the B cells, these are our big guns that are searching for targets. We want to make absolutely sure that we are not the target. When they start attacking our own body cells, you get diseases, rheumatoid arthritis, lupus, other diseases like that. It is very hard to treat those. So we need to make sure first that the T cells are functional and they can do their job, and second that they don't attack us. That's the whole purpose of what's happening in clonal deletion. All right, vital that immune responses not be against autoantigens. Remember, we call our proteins that just our regular proteins, if an immune cell is going to bind to it, we refer to that as an autoantigen. We don't want that to happen. So we have to eliminate those. Okay, so let's talk about how this occurs. All right, so there's my stem cell in my red bone marrow. I've made some T cells. They have migrated to the thymus. They all have their T cell receptor. You can see that that antigen binding site is unique, how it's shaped differently. It was designed to bind with a soulmate antigen. Now that variability just came from picking 3 different parts and putting them together. You remember it took from the DNA, took a little bit here and a little bit there and a little bit there, put it together, and we compare that to making outfits, and you grab a pair of a shirt, a pair of pants, and some shoes, and put them together. Now we know, ladies, we know that some of those outfits don't work. Just because I can put them together doesn't mean that they should go together. So clonal deletion is all about weeding out the T cell receptors that either don't work or they're going to respond against autoantigens, respond against self. All right, so the first question that happens in the thymus, and I have all these immature T cells coming in, is will it recognize that MHC protein? Thymus is a cell and it's capable of producing class 1 MHC proteins bound to different epitopes. So the first thing we need to ask is, can it actually bind to MHC? And you remember the T cell receptor not only is looking for that soulmate antigen, it is also looking to see if it can bind to MHC. So that's the first question. Will my T cell recognize MHC? If it can, then it can do a job. It can do a job. So yes, it can do a job. We're going to take it along further. If it can't, then this T cell will not do the job. It will not be able to recognize this, bind to it, and start an immune response. No reason in keeping employees that don't work, so this T cell can't do its job, cannot bind to MHC, so we're going to trigger it for cell death. That's that apoptosis. So this one is out. Will it recognize MHC? No, it can't do a job as a T cell. Get rid of it. All right, this one can recognize MHC, so that's good. The next question we need to ask is, will it recognize our own autoantigens, our own body's protein? Now, this is what's cool about the thymus. The thymus has a job, but also one of its job is it's able to present proteins that are used in all different cells. Okay, so if you remember right, all of our cells contain a complete copy of our DNA. We've got all of our instructions in every single one of our body cells. But an epithelial cell only needs to make proteins for epithelial cells, and a heart cell only needs to make cardiac proteins that are involved with the heart. And so the proteins, even though they have the instructions for them, they don't make them. The thymus makes proteins from all our different instructions. It has the ability to make pieces of proteins from all of our body's instructions. That's what's cool about the thymus. That's what these T cells are looking at. These T cells will look and see if it matches any of these proteins from any cell in our body. And the thymus has the unique ability to present those to that. So here's our T cell. This particular T cell matches with one of my own body's antigens. Remember, we're not talking about foreign invaders right now. We're just checking to see if it will attack our own body. And look, this one does. If it attacks our own body, yes, that's a problem. We got to get rid of that guy. Apoptosis. So if it recognizes MHC, it can do its job. That's good. But if it recognizes a autoantigen, that's bad. So this cell is going to be marked for apoptosis. Now there is a few exceptions. Our body actually keeps a few of these, and they actually differentiate and become regulatory T cells. And for some reason, these regulatory T cells help train our body, and they help prevent autoimmune diseases. And when our body doesn't make these regulatory T cells correctly, then we're more susceptible to some of these autoimmune diseases like lupus and others. So that's kind of interesting. Don't exactly know how that works, but we do know a few of these, even though they recognize autoantigens, are kept, and they become our regulatory T cells. All right, so let's talk about the others. There are T cells that work. They recognize MHC, but then they don't recognize any autoantigens. So if the answer is no, they don't recognize any of our own body antigens, these are the guys that we want. These are the guys that have a soulmate antigen that's not present in our body, and if that soulmate antigen and comes into our body, is a foreign body and comes in, this will recognize it and kill it. So these are going to become our T cells that are ready to go. Now this book for some reason calls it immature T cells. It's not consistent in the literature. Some people call this mature T cells. They still need to undergo activation, but these cells are ready to go. Will move into our our circulatory system and our lymphatic system, and they will start doing their job. And their job is to look for the soulmate antigen, and if that happens, then their job is to kill, and get rid of it. So this is called clonal deletion. The reason it's called clonal deletion is because when I get rid of a cell that doesn't work, or when your body gets rid of a cell that's going to bind to an autoantigen, not only does it get rid of that cell, but it gets rid of any future progeny of that cell. It makes sure that any clones of that cell, because I got rid of this one, then no clones will ever be done. So that's why they call it clonal deletion. You're deleting the ones and any potential clones that could have come from that. All right, so this is clonal deletion of T cells. Do know that B cells will go through a similar process in the bone marrow, but this is very important in our overall ability. We need T cells that work, that recognize MHC, and then we need T cells that do not recognize autoantigens, and that's how we get our repertoire of T cells. Okay, so that's it for clonal deletion. That can be tough. It's not too bad. Go through it a couple of times with the video, talk it out, and then we can talk about it in Zoom too. Thanks.